In this video, we're going to look at how the cells can repair their damaged DNA. There are 7 ways cells can repair the damage to DNA, and the method used depends on the type of damage to the DNA. The first 3 methods I'm going to talk about deal with DNA damage where there is typically a chemical change to a base such that it is no longer a usual base you find in DNA. These methods are direct repair to reverse the DNA damage. This method is not often used. Base excision repair, which is the primary repair method used by cells and can deal with single damaged bases and single strands breaks, which are also called NHEJs. Nucleotide excision repair, which is when the cell removes thymine dimers caused by UV damage and also deal with large chemical adducts on DNA. The next 2 methods can deal with errors during copying, and these are the proofreading activity of the DNA polymerase and mismatch repair, which removes the mismatched base pairs and fixes insertions and deletions. Finally, the cell has to deal with damage to the sugar phosphate backbone, where the bonds have been broken on both strands to give what we call a double-stranded break. For this, the cell has 2 mechanisms: non-homologous end joining and homologous recombination repair. We will now have a look at some of these mechanisms in a little more detail. First, base excision repair, which is the primary repair method for damaged DNA and which can deal with single damaged bases and also single strand breaks. The way this method works is relatively straightforward. The damaged base is identified and removed, and then replaced with the correct base using the other strand as a template for the correction. The damaged base is recognized by an enzyme called DNA glycosylase, which removes the damaged base. Next, AP endonuclease and a phosphodiesterase will cut the sugar phosphate backbone of the DNA, and then DNA polymerase B inserts the correct base. Finally, the nick is sealed by DNA ligase. When multiple bases are involved, the cell will use nucleotide excision repair. In nucleotide excision repair, a nuclease will cut a 24 to 32 nucleotide region that spans the area of damage, and then a helicase will open up the DNA so the cut segment can be removed. The missing DNA is then replaced using DNA polymerase E or D, and finally the nick is sealed with DNA ligase. It's interesting to compare and contrast these 2 excision methods. They both use different enzymes. One removes one base, the other removes many bases, and both use different polymerases to repair the error by using the complementary strand. And they both use ligase to finish off the process. The methods are the same, but also different. Now moving on, let's have a look at what happens when an incorrect base has been inserted during copying or missed out altogether. The cell copies its DNA at a reasonably fast rate, and during this copying process errors can occur, and the cell needs to correct these errors. To correct the DNA copying mistakes, the cell can use one of 2 methods. It can either use the proofreading properties of the polymerase doing the copying, or it can use mismatch repair. In mismatch repair, the site of mismatched bases is recognized, and this causes the recruitment of mismatch repair factors that in turn recruit an exonuclease to remove the mismatched base, and also some of the surrounding bases. Next, a DNA polymerase replaces the removed bases, and finally a DNA ligase comes in and repairs the nick. Now, one important question here is, how does the cell know which is the correct base and which is the incorrect base, or what is an insertion or what is a deletion? Well, in bacteria, the old parent strand can be identified as it contains added methyl groups, whereas the new strand will not. However, in eukaryotes, the mechanism is not known. Finally, let's look at how cells deal with double-stranded breaks to the DNA backbone. The cell has 2 methods for dealing with double-stranded breaks: non-homologous end joining and homologous recombination repair. In non-homologous end joining, the double-stranded break is repaired by rejoining the 2 broken ends, regardless of the sequence. This approach is obviously error-prone, as the wrong ends may be joined if there are multiple breaks, or some loss or gain of DNA sequence may occur. In homologous recombination repair, which is similar to homologous recombination in meiosis, you need the presence of the sister chromatid, the other chromosome, as it will act as a template to help the repair. In homologous recombination repair, a nuclease processes the 5' end of the break, then a single strand of DNA from the donor sister chromatid invades the damaged region and acts as a template for the repair. This means the repair is more accurate than in non-homologous end joining, as there is a copy of the DNA to work from. Now the cell must deal with damage to its DNA, because if it doesn't, then it may lead to the cell not functioning correctly. In fact, as part of cell division cycle, the DNA will be inspected for damage and the cell cycle will be stopped until the damage is repaired or the cell determines that the damage can be tolerated. So, in DNA repair, there are some common themes between the different repair mechanisms. Proteins detect the damage and bind, the damage is removed, often through the action of nucleases, then DNA polymerases and ligases repair the damage. The whole process is usually regulated by a group of proteins called protein kinases. In terms of outcomes, if we get an accurate repair, then the cell survives. If the repair is not successful, then we get cell death. If there is a misrepair, then we get a mutation and a loss of genome stability. So, in this video, we have had a look at the various methods that a cell can use to repair DNA damage.